There we go. So when we're talking about mitosis, we're talking about reproduction of the cell exactly. The resulting cells are just like the original. They have the same number of chromosomes. They're using the same information in the same way. So you have a reasonable expectation that they're going to look and act like the original cell. Okay. So we start off with a discussion of cancer, right? What is cancer? Cancer is uncontrolled cell growth. So cancer is actually not so much a disease as it is a symptom. The symptom is uncontrolled cell growth. The cause is mutation, all right? Mutation of what? Usually a gene involved in the cell cycle. Remember that when the cell divides, we have mitosis, prophase, metaphase, anaphase, telophase. And then we follow that with interphase, right? And interphase is broken up into G1S and G2, right? In G1, we have a gap before we synthesize new DNA, all right, getting ready for mitosis. In G2, we have a tiny gap before we divide, all right? Now, some cells are constantly going from G1 to S to G2. Some cells go into G1 and then from there to G0, and they don't divide for the rest of your life. It all depends on the type of cell, right? The switch from mitosis to interphase, back to mitosis to interphase, is governed by a whole bunch of genes that control when and where the cell is going to do its thing, all right? And so that involves stuff like knowing of what's going on around you, being able to sense the presence of things like growth factors in the environment to tell you to divide, okay? Or being able to pick up certain cell products nearby that tell you you need to replace what's been destroyed, okay? So in cancer, one of those genes gets messed up, okay? It either makes too much or too little of the product needed to control the cell cycle. And the result is that the cell divides and divides and divides without paying any attention to the need of the host. Now, sometimes these cells can migrate throughout the body, through the blood and through the lymphatic system, and sometimes they stay where they're at, all right? When they're able to migrate, that's called metastasis. And they can go from what's called their primary focus or the primary tumor to setting up secondary tumors wherever they land, okay? In a benign cancer, the cells just reproduce in one spot and don't move, okay? An example of that would be something like a brain tumor, a glioma, okay? Whereas a malignancy could be in any of a number of tissues in the body, generally epithelial and or connective tissue, because they divide frequently, whereas other types of tissue like muscle and nerve tissue don't, okay? One of the hallmarks of cancer is that once a cell becomes cancerous, it never dies. It's immortal. So it just divides. As long as you feed it, it keeps going. It usually generates chemical factors that provide a blood supply to it, so it never dies. And as a result, it's stealing from healthy tissue what it needs to live. The oxygen, the nutrients, the good stuff all goes to the tumor. And so you literally suck the life out of the host, okay? An example of a tumor cell line is the HeLa line, named after Henrietta Lacks, who died of that particular cancer. But we've used her cell line many times over for research into how cells and how cancer cells work, okay? Understanding why cancer cells are immortal and why we're not begins the understanding of what it is that keeps us alive and how we die. And we've talked a little bit about that already, right? We talked about the immortal baby, and we talked about progeria earlier when we were talking about cells, right? And one of the things that we talked about was the fact that in order for the cell to retain its information, it has to complete the tips of the chromosomes using an enzyme called telomerase. And if it doesn't do that, with every cell division, it loses a little bit of information until you get to the point where you lose critical information and the cell says, I give up, and then it dies. And so the reason we die, one of the reasons we die, is that the rate of cell death exceeds the rate of cell replacement. And when that happens, tissues thin, organs begin not to work as well, organ systems shut down, and then that's it. You're done, okay? So that clock is pegged to how fast those cells reproduce and how well the information in the nucleus is maintained, because that's critical for the cell to survive. There's other factors as well, right? This is just showing you a picture of Henrietta Lacks and a micrograph of the HeLa cell. Again, as long as you feed it, it never dies, okay? But it doesn't do anything for the host except remove vital components in the blood that it needs to survive, right? So it only serves itself, okay? That's why I always tell people that cancer is a symptom and not a disease, right? The symptom is uncontrolled cell growth. The cause is mutation, okay? It would be kind of like you going into the doctor's office and the doctor said, "What's wrong?" And you say, "I'm bleeding." Well, okay, I can see that, but what we need to figure out is why, okay? And if we can figure out why you're bleeding, then we can make the symptom go away, right? It's the same thing with cancer. If we can figure out why this thing is growing, then we can make it go away, okay? This is a little bit on mitosis and meiosis. It kind of goes through each. It's a little bit old fashioned, but I like it because it's thorough. So let's take a listen. You talk? I don't think you talk. You're not even running. Here we go. You running? You're not running. All right, let's do this. Okay. Yeah. Add into speakers. One. And here.
Hi.
Yeah, I'm here. You're here. Let's see. Let's go to. Correct. We can go to. I want to go to. Let's do this. And let's see what's here. Let's go to platform. Moving to it. Okay, you got that. You got the output. Yes. We're coming. Now we can share. This is an oak tree. It doesn't look much like one, but it is. A small acorn has the complete potential to become a 8-foot-high oak tree, because within the acorn is the blueprint for every cell that oak tree. In the living process of growth, the cells of the seed are duplicated billions upon billions of times, guided unerringly by the blueprint of its genetic material, resulting in the transformation of that seed into a tree. And the essence of the growth process is the same for virtually all living organisms on Earth.
own special blueprint for each species. Now, the amazing thing is that the blueprint for every single living creature on Earth is composed of the same kind of molecule, which scientists know as DNA. The DNA is organized into tiny subunits called genes. Genes are responsible for producing the various traits of an organism. DNA does this by supplying the information needed by cells for the manufacture of the proteins that make up all living substance. The alphabet of life. Just as there are few words in English that consist only of a single letter, most traits in the organism are the result of the interaction of lineage. For example, each book you see, yet every one of them is written with words made up of letters from the same alphabet. The number of meaningful stories that could be written using different combinations of the same letters is almost infinite. The different organisms we see around us are the different books of life that DNA is used to write. Organisms grow by the duplication and division of the cells within them. Mitosis is the method by which nearly all such cell division occurs. It is the means by which the master plan of the DNA is passed from cell to cell in a growing organism. If we watch mitosis in action, as in the single-celled animal, it is easily seen that the result is the formation of two identical cells from an original cell. Watching cells divide, we should try to be aware of their three-dimensional nature, as these scanning electron micrographs of mitosis illustrate. Healthy young salamander is undergoing rapid growth to maturity. One would expect its cells to be in a constant state of division. However, even in the salamander's most rapidly growing tissues, the cells divide only about twice a day, and most cells divide even more slowly. For the greatest portion of their life cycle, cells are in a state known as interphase. During interphase, the DNA formed in the nucleus is distributed as an indistinct granular substance known as chromatin. While biologists used to think that interphase was a resting period for a cell between divisions, we now know that nothing is further from the truth. Although it isn't obvious, the cell during interphase is a dynamo of activity. During this time, the cell is engaged in duplicating all the structures and materials that the new cells will need following division. Among these, perhaps the most important is DNA. Process of DNA replication occurs near the middle of the interphase period. It's important to remember that chromosomal DNA is replicated only during interphase. It's a process that does not occur during any other stage of a cell's life cycle. This replication of DNA must be absolutely exact, so that the two identical copies of the genetic material can be passed on without modification to the two new cells that will be created in the coming division. After replication is completed, the DNA, shown here as a simple chain, begins to coil upon itself. And while proteins are associated with this coil, they are not shown so that we may watch more closely what is happening to the DNA. As we move through the DNA molecule, we reach a point called the centromere, where the molecule is joined to its exact replicate. Together, these two DNA molecules form a chromosome. The appearance of chromosomes marks the visible beginning of the division process. As the chromosomes appear, the nuclear membrane disappears. Telomeres cell. The chromosomes now migrate to the center of the cell, aligning themselves along its equator. The centromere of each chromosome splits, and each half of the original chromosome now becomes a chromosome containing only one molecule. The has moved to opposite sides of the cell as the cell membrane pinches in, forming two new cells. Each of these new cells now forms a nucleus containing the same number of DNA molecules, identical in structure to those in the original cell. The division we have just seen actually took about 20 minutes. The two new cells formed in mitosis now enter interphase and will not be ready to begin division again for another 24 hours. However, not all cells are destined to keep dividing over and over again. Human nerve cells stop dividing shortly before we are born, and if they are lost, can never be replaced. Other kinds of cells will only divide under special circumstances. Liver cells, for example, never divide in the adult organism unless the liver has been damaged, and cell division is required to repair the organ. During the interphase, the cell duplicates its DNA. The chromosomes become visible from the DNA coil. Chromosomes align themselves in the center of the cell. The chromosomes then split and move to opposite sides of the cell. The division of mitosis results in two cells, both identical in composition to the original cell. Cell division that makes possible the fundamental life process of sexual. In sexual reproduction, the cell from the male of the species is combined with the cell from the female of the species, resulting in a single cell, a zygote. Complete organism, accomplishes this by creating a new kind of cell, sex cell. DNA found in the other body cells of the organism. For example, humans have 46 chromosomes. 23 of these come from the mother and 23 from the father. Meiosis makes this possible. Meiosis reduces the number of chromosomes in half, in this case from 46 to 23. So when a sperm cell from the father fuses with an egg cell from the mother, the result is a single fertilized cell, which again contains 46 chromosomes. Meiosis begins with a cell in the sex organs, in interphase, and closely resembles the beginning of mitosis. It is only when the chromosomes begin to form that we can see a noticeable difference. A remarkable thing begins to happen. Pairs of lookalike chromosomes approach each other and join along their entire length. The chromosomes come so close together that they overlap each other. Genetic material is exchanged during this union between the points at which the chromosomes overlap. Exchange of segments in this twisting and tangling of chromosomes is called crossing over. Mind the fact that each chromosome can carry thousands or more genes, we can begin to appreciate the staggering number of genes that can be shuffled during crossing over. Lookalike chromosomes only occurs in meiosis. crossover can be easily observed in living cells, and it's one of the most visible differences between meiosis and mitosis. In the salamander cell, crossover is occurring between pairs of look-alike chromosomes. These figures created by the chromosomes during crossover are called chiasmatic. After crossing over, the chromosome pairs partially separate and migrate toward the center of the cell. There, they separate completely. Unlike mitosis, these chromosomes still contain two DNA molecules. The cell membrane then pushes in, forming two new cells. Each of these new cells now enters a stage that is reminiscent of interphase, except that no new DNA is replicated. The chromosomes again migrate to the center of the cell. The centromere is split and the chromosomes separate, each half moving to the opposite sides of the cell. These new chromosomes only contain one molecule of DNA, so that the four cells that now form have half the number of chromosomes and half the number of DNA as the body cells of the organism. Watch meiosis actually happen in a salamander. As the chromosomes begin to appear, crossing over is occurring. The chromosomes partially separate and then align themselves in the center of the cell. They then separate completely and two new cells form. While in salamanders a nucleus forms and the chromosomes disappear, in some organisms no nucleus is formed and the chromosomes remain visible. Even though this pause strongly resembles interphase, it's important to remember no DNA is replicated in this stage of the cell's life cycle. The second meiotic division is marked by the reappearance of chromosomes. Cells about to undergo the second division, chromosomes migrate directly to the center of the cell in a manner reminiscent of mitosis. Once there, they separate, and the second division soon follows. The four cells that result from the two divisions of meiosis have half the number of chromosomes and half the amount of DNA as had the original cell previous to interphase. These new cells now develop into sperm, which are able to fuse with an egg cell, restoring the chromosome number to that of the body cells of the organism. During interphase, the cell replicates its DNA. Over begins when look-alike chromosomes pair. After crossover is complete, the pairs partially separate and line up in the center of the cell. Splitting occurs with each chromosome still containing two DNA molecules. the genetic composition of these molecules has been changed. Two new cells are created by the first division, each with DNA still equal in amount to the organism's body cells. The cells enter a resting period during which no DNA is replicated. Chromosomes then reappear. No crossover occurs during this time. After splitting, each chromosome has a single molecule of DNA. At the end of the second division, each cell has half the amount of DNA and half the chromosome number as the original cell had before entering interphase. Mitosis and meiosis appear at first glance to be remarkably similar. In both mitosis and meiosis, the cell enters interphase with 4 chromosomes, each of which has one DNA molecule. In both cases, the DNA is doubled during interphase, resulting in cells that have the same amount of chromosomes but twice as many DNA molecules. In mitosis, the chromosomes line up in the center of the cell. In meiosis, look-alike chromosomes pair and crossing over occurs. In mitosis, the centromeres split and the new chromosomes contain one DNA molecule. In meiosis, the chromosome pairs partially separate and line up in the center of the cell. In mitosis, division occurs. Each of the new cells has the same number of chromosomes and the same amount of DNA as did the original cell. In meiosis, the chromosome pairs separate after they have exchanged genetic information. Each chromosome contains 2 DNA molecules. After division, meiosis continues as the chromosomes migrate to the center of the cell. The centromeres then split and the chromosomes move to opposite sides of the cell. Each chromosome now has only one DNA molecule. Each of the 4 cells formed has half the number of chromosomes and half the amount of DNA as did the original cell.
Okay. So
I cannot find that video.
I'll post it,
Okay.
but it's also embedded in the presentation. Yeah. Yeah. So again, right, at the end of the entire process, right, what do we have? So share the screen back here again. We want that. And that. Oh. Okay. At the end of mitosis, you have 2 new cells with the same number of chromosomes, no rearrangement of DNA. They're each getting the same information, and they're going to look and act like the original. Okay. So, for instance, in a human cell with 46 chromosomes, when that cell divides by mitosis, each new cell is going to have 46. Okay. No change in the amount of genetic information happens in the daughters. All right. In meiosis, it's different. Okay. In meiosis, you end up with 4 cells, each with half the amount of genetic material as the original. That genetic material has also been recombined in 50% of the cases, which means that there's a unique arrangement of genes that's not present in the parent. All right. And this is how we make eggs and sperm. Okay. When they come together in fertilization, what happens? You get an egg cell with 23 and a sperm cell with 23. They come together and fuse and they make a new cell, a zygote with 46. And then we can build a new organism, a new human being, for instance. Okay. A cell reproduces by dividing. Each descendant receives a full set of chromosomes and some cytoplasm. Before the cell's nucleus divides, it reproduces its chromosome. In mitosis, we're using this for growth and repair and maintenance. All right. So this is an example of what you would call asexual reproduction, right? There is no male and female to it. There is simply cell division. Okay. So here's a look at asexual reproduction.
One of the most intriguing uses of stem cells can be found in the field of regeneration. Whether the goal is to regenerate damaged heart or nerve cells, or to regenerate an entire limb, a similar process must occur. Damaged tissue must rewind development, calling on cells that can reverse their differentiation, or rallying reservoirs of stem cells that can generate both differentiated cells and more stem cells. Humans can regenerate certain parts of the body. Wound healing is a poor cousin of regeneration. Human liver, for example, can regenerate an entire piece if it has been lost through damage or surgery. Other vertebrates are more proficient than mammals at regeneration. Some lizards and salamanders regenerate lost tails, and salamanders can regenerate entire limbs. The organism with one of the most incredible abilities to regenerate is the pig. Worms' regenerative capabilities have been known for hundreds of years. And today, modern molecular biology techniques are turning this old favorite into a powerful new model for understanding the potential of stem cells. Planarians are free-living flatworms and are related to parasitic flatworms like the liver fluke and the tapeworm. They are found throughout the world in wet environments. They range in size from 3
Hello. What happened? Wake up.
Sometimes.
Must be. Hold on a second, ladies and gentlemen. Here we go. Play. Come on. Wake up. Oh. Let's see if you got it again. There we go. Yeah, we back. All right. Okay. Share. There we go. We got that. Okay. Now, my point was when I was talking earlier about mitosis versus meiosis, right? Meiosis is for sexual reproduction, okay? Meiosis produces haploid gametes that have rearranged DNA, unlike either parent in 50% of the cases. And as a result, that increases the biodiversity of the population, all right? Now, with mitosis, what you're doing is you're generating an identical copy of the original cell by keeping the amount of DNA the same and by not rearranging any of it, okay? So when you have a population that reproduces asexually, you don't have a lot of biodiversity because everybody's a carbon copy of everybody else, all right? In the body, in the human body, for instance, different ways to reproduce and maintain and repair tissues include fibrosis and regeneration. Fibrosis is scar tissue formation. Regeneration is producing the exact same tissue from the previous tissue, all right? In the human body, the tissues come into four types. There's epithelial tissue, there's muscle tissue, there's connective tissue, and there's nervous tissue. Muscle tissue and nervous tissue do not regenerate and do not fibrose. They simply, when you lose it, you get scar tissue back, okay? So I guess you could say they fibrose to an extent, but you don't get the nerve cells back and you don't get the muscle cells back. They're gone for good. In epithelial tissue and connective tissue, you get regeneration. So you get replacement of the old tissue with new tissue that's very similar to it, okay? Provided you don't destroy the stem cells that generate it, okay? So that's the difference, for instance, with a deep cut versus a shallow cut, right? A shallow cut will grow back and it'll look just like the original skin over the top, right? But a deep cut that goes down below the epithelial layer will produce a scar, right? Everybody has scars on their body. And if you look at a scar, you notice that it's different from the surrounding tissue. It doesn't have hair follicles. It doesn't have sweat glands. It doesn't have oil glands in it, okay? And in many cases, it doesn't have nearly as many nerve endings, all right? And that is just a way of saying that what we're doing here is not replacing the tissue. We're just doing a patch job, okay? When you have too much scar tissue formation, either inside or outside the body, you lose function, okay? You can scar inside your body and outside your body as a result of trauma or disease, okay? But when we're talking about using it for reproduction, all right, we're talking about making a clone of the original cell, all right? So one of the organisms where that happens is our friend, the planarian. You can catch planarians around here with a piece of liver and a fish hook and some dental floss. Just find a nearby puddle. It doesn't matter where it is. And you leave that liver in there overnight and you'll pull out some planarians the next day, okay? And then you can do these kind of experiments at home. You can take that planarian, cut him in half, and he'll regrow. You'll get two worms back. And why is that? Because he has a stem cell population that can reproduce the entire organism, okay? This is one of the unusual examples of a multicellular organism that can reproduce asexually, okay? Yeah, he does mitosis. So most asexually reproducing organisms are single-celled, like bacteria, or single-celled eukaryotes like protozoa, right? They just divide, right? Most multicellular organisms reproduce sexually, right? But this is an example of a multicellular organism that can do it asexually, okay? And that's why it's of interest for research. So let's take a listen. You gonna work for me? You gonna go, baby, or not? Hold on. Let's go one more time. Get back up and then let's go. All right. You're gonna argue with me. Let's do this. Decide where it needs to be. Good. Should be good. All right, so do it.
One of the most intriguing uses of stem cells can be found in the field of regeneration. One of the most intriguing uses of stem cells can be found in the field of regeneration. Whether the goal is to regenerate damaged heart or nerve cells, or to regenerate an entire limb, a similar process must occur. A damaged tissue must rewind development, calling on cells that can reverse their differentiation, or rallying reservoirs of stem cells that can generate both differentiated cells and more stem cells. Humans can regenerate certain parts of the body. Wound healing is a poor cousin of regeneration. The human liver, for example, can regenerate an entire piece if it has been lost through damage or surgery. Other vertebrates are more proficient than mammals at regeneration. Some lizards and salamanders regenerate lost tails, and salamanders can regenerate entire limbs. However, the organism with one of the most incredible abilities to regenerate is the planarian. This worm's regenerative capabilities have been known for hundreds of years, and today, modern molecular biology techniques are turning this old favorite into a powerful new model for understanding the potential of stem cells. Planarians are free-living flatworms and are related to parasitic flatworms like the liver fluke and the tapeworm. They are found throughout the world in wet environments. They range in size from 3 to 12 millimeters and are scavengers, feeding on the decaying remains of animals and plants. Dr. Alejandro Sanchez Alvarado, an HHMI investigator at the University of Utah, describes some of the planarians' physiology.
Planarians derive their name from the fact that they're very flat. Planet mean flat and hence planarians. And the reason why they're interesting is because these are perhaps the simplest organisms that display a centralized nervous system. So the central nervous system is composed of two lobes that look like a horseshoe shape around this area right here. And those two are connected to ventral cords that send projections all the way down to the very tip of the animal. And this is how the brain basically controls the motion and musculature of these animals. This dark gray area right here is part of the gastrovascular system. This little thing right here is a muscular pharynx, which actually comes out from the ventral surface, from the belly of the animal out, and is used to literally impale food and suck it back in. Now, planarians are funny because they don't have a mouth, so they only have a single opening to their body, and it's through this pharynx. So the food enters through the pharynx, it is digested, and whatever is not digested and is not dumped into the rest of the animal comes back out through the same pharynx. So it's an organ that serves two functions. These two cartoon-looking cockeyed eyes right here actually are the photoreceptors of these planarians, also known as eye spots. And what these eye spots can do is detect light. And planarians don't like light. They're phototactically negative. So whenever they see light, they'll just run away from the light and they'll hide somewhere. And this is perhaps, the prototypic, the winged eye, which consists of a light-sensing organ and a pigment cup organ that allows for the light to be handled by the organism.
But what makes a planarian so exceptional at regeneration is its ability to rapidly grow even very small body pieces into completely formed worms. Unlike the cells of most animals, most of the cells in a planarian's body can act like stem cells. If you cut a planarian in half between its head and tail, the tail half grows a head and the head half grows a tail. If you cut it lengthwise, the two halves grow new second halves. You can even bisect just the head between the two eye-like photoreceptors, and you will end up with a worm with two heads. This process can be taken to incredible extremes. Dr. Sanchez Alvarado describes the planarian's ability to regenerate.
So you can take a planarian and slice it into 8 fragments, okay? And each fragment will go on and regenerate a complete animal. That would be like saying that you can cut your ear off and then a new you will arise from that ear. Well, planarians can do that. We can, but planarians can do that. Morgan in 1898 demonstrated that you can take a fragment that's 1/279th the size of the original animal, and the tiny little fragment for the flank of the animal will be capable of restoring all of the missing parts and produce a complete and properly shaped organism. And it does this in about a week or so. And so that just gives you an idea of how plastic these animals are.
Thomas Hunt Morgan investigated the planarian's regenerative powers at the turn of the 20th century. Even before Morgan, Charles Darwin wrote about planarians he found during his voyage on the Beagle, describing their abilities and illustrating their anatomy. What new information has modern-day biology been able to uncover about planarian stem cells and regeneration? And what do these findings tell us about the human capacity for regeneration? At the University of Utah, the Sanchez Alvarado Lab is using today's molecular biology tools to uncover the secrets behind the planarian's remarkable abilities. They use a particular type of planarian, Schmittia mediterranea, in their research. Otto Rudelhofer, a graduate student in the Sanchez Alvarado Lab, explains the advantages of using planarians to study stem cells.
But I didn't feel like the mammalian system is ready to tell us a lot. I felt like a lot of the basic knowledge that we need to know in order to ask the right questions about stem cells needed to be discovered in a much simpler organism. So although the mouse work is very, very interesting, I thought studying it in a much simpler organism would be much more interesting. And that's how I got turned on to studying stem cell biology in planaria.
Experiments using RNA interference, or RNAi, were designed to identify the genes involved in the planarian's neoplasts, the stem cell-like cells that migrate to areas of damage and allow the worm to regrow the missing parts of its body. The RNAi experiments identified 240 genes involved in a planarian's regeneration, including one coding for a protein called SMEDRI that is similar to proteins found in fruit fly stem cells and during the process of human sperm maturation.
This molecule, SMEDRI, receives its name because it's a homologue of a molecule that was discovered in Drosophila called PIWI. And PIWI is a molecule that was identified in Drosophila as being responsible for the maintenance of the stem cells in the gonads of the female fly. We were expecting PIWI to actually play a role in regulating the maintenance of the stem cells that are found in the body plan of planarians. And so the original phenotype, the defect that we uncovered by eliminating these changes in the RNA RNA, said that was the case. Because the animals look a lot like what irrigated animals look like. And irrigated animals basically have no stem cells, because the stem cells are the only cells that divide in planarians. So you subject them to large doses of irradiation, like you would do for cancer, for example, all the dividing cells are eliminated. And there's no stem cells to produce new daughter cells to go in and replace these dying cells, and the animal dies. And it dies in a very specific way. The hair begins to resort back, and the body begins to assume a ventral curling. So the animal now looks almost like a taco shell. When we science people with RNAi, we got exactly the same phenotype, a regression of the head and its current phenotype. So we thought, aha, when we look at these animals in detail, all those stem cells are going to be gone because we're essentially getting the same defect. But we were surprised because when we went and looked at these animals, we realized that the stem cells were there. They were happy. The stem cells were actually responding to tissue turnover, so they were producing division progeny. And so the message there was to us that pinwheel not only had a function in the maintenance of stem cells, but it also had a function in regulating the function of daughter cells. So you're causing a defect early on in the life history, even before that cell is born, that when the cell is now born, it's supposed to go on and differentiate, it cannot. And then it fails and it undergoes apoptosis most likely, and then it dies. So the lesson is actually quite profound for us because it tells us that this molecule PME is actually doing something to the genome of the stem cell that will determine whether or not the daughter cell will be functional.
But how much of this knowledge will be relevant to human stem cells?
I think the main thing that could be applied to human invertebrate biology based on what we're learning in the planaria right now is identification of genes. So by identifying these genes that are involved in planaria regeneration, hopefully we can understand their function in humans. These are genes that are promoting regeneration in planaria. It may be possible somewhere far down the line to turn these genes on pharmacologically, and they may have some effect in vertebrates.
Multicellularity has been around for about 600 million years. So the history of life on this planet is unicellular. It's just all single cells. So it took, 3.5 billion years, only 600 million years ago, multicellularity emerged. And one of the fundamental attributes, I think, of multicellularity is the maintenance or segregation of the germline into stem cells to repair tissue, which we damage all the time. Even Drosophila has now stem cells in its gut, which is absolutely required to keep nutrition to the stem cell in the gonad such that the animal can reproduce. So I think all these processes at some level are going to be conserved, and by studying planarians, we might be able to inform that biology, but by extension, understand our own biology a little bit better than we do today.
So the lesson there, right, is that when you're talking about asexual reproduction, you're talking about recreating the original tissue by maintaining the same amount of genetic material and using it the same way. What they did in the video was ask the question, what makes a stem cell a stem cell? A stem cell is a cell that can become any type of tissue that it wants to, and it does that through a process of changing the genes that it uses to define what its job is, right? It eventually only reads from the DNA handbook that's important for its function. Like a muscle cell only reads from the muscle cell handbook of DNA, and a skin cell only reads from the skin cell handbook. The other information is still there, but it's not used for transcription and translation. And so that changes the way the cell looks and acts, all right? And so what they were doing was trying to figure out what the role of certain genes was in maintaining the stem cell population and what the effect was on the progeny cells, right? In interphase, that's where the cell does most of its job, right? In the eukaryotic cycle, the interval between mitotic divisions, when a cell grows, roughly doubles the number of its cytoplasmic components and replaces its DNA. The three stages are G1, S, and G2. We talked about those earlier. G1 is gap1, and that's an interval before DNA reproduction. S for synthesis of DNA. And then G2 is gap2, when the cell prepares to divide. And then we have mitosis, right? Prophase, metaphase, anaphase, telophase, and then cytokinesis in most cases, right? So this is just a figure showing you the eukaryotic cell cycle, right? A cell spends most of its time in interphase, right? G1 is the phase of growth before DNA replication. The cell's chromosomes are unduplicated here. S is synthesis phase, where we make copies of the genetic material by DNA replication. G2, the phase prior to mitosis, where it prepares to divide. And in the following set of phases that include mitosis, right, the nuclear material divides, right? The chromosomes condense, they line up at the metaphase plate. At anaphase, they're pulled apart. At telophase, they arrive at opposite ends of the cell. the nuclei reform, and then the cell pinches in half, and that's cytokinesis, okay? You end up with two cells looking and acting just like the original cell, all right? In many cases, the cell then repeats that cycle, right? Goes back into interphase and then back into mitosis and over again. But in some cases, the cell does not, okay? And we talked about, for instance, in muscle tissue and nerve tissue, that doesn't happen, right? You get all the cells you're going to get, and then as you grow, they just die off, okay? So the cell cycle is just a series of events from the time a cell forms until the cytoplasm divides, including interphase, mitosis, and cytokinesis, right? Mitosis distributes the complete set of chromosomes into two new nuclei. In G2, each chromosome consists of two replicated DNA molecules attached to the centromere. When the sister chromatids are pulled apart, each becomes an individual chromosome in a new nucleus. When the cytoplasm divides, the two nuclei are packaged into two separate cells, okay? So you can see here, right? Prior to DNA replication, each chromosome consists of one DNA molecule and protein. Those are the histones and the non-histone proteins. One chromosome of each homologous pair is inherited from each parent. After DNA replication, each chromosome consists of two molecules of DNA attached to sister chromatids. Mitosis and cytoplasmic division separate the sister chromatids of each chromosome and package them in new nuclei. Each new cell has the chromosome number of the parent, okay? So mitosis is division of the nuclei, and cytokinesis is the division of everything else in the cell, okay? Homologous chromosomes are chromosomes with the same basic information on them, all right? You get a pair of each chromosome, one from your mom, one from your dad, all right? We have 23 chromosome pairs. Each pair of chromosomes is basically the same, but not identical, okay? Except for the pairing of the sex chromosomes in males, the chromosomes of each pair are about the same size and have approximately the same information on them, okay? One member of each homologous pair was inherited from the female and one from the male. And you can see here, again, mitosis and cytokinesis. On the left, you've got a cartoon representation of that. In the middle, you've got actual electron micrograph of an animal cell dividing, okay? And on the right, you have, not an electron micrograph, sorry, just a micrograph. And on the right, you have a micrograph of a eukaryotic cell dividing with the chromosomes stained so that you can very easily see the different phases of mitosis, right? Prophase, metaphase, anaphase, telophase, and then back into interphase, right? We start with interphase, right? Interphase cells are shown for comparison here, but interphase is not part of mitosis. The nuclear envelope is intact, and the loosened chromosomes are not easily visible under a light microscope. DNA replication occurs before mitosis begins, so each chromosome consists of two sister chromatids, right? So what are we doing here? We're generating new DNA, and we're getting ready for the division dance, okay? Or, you know, if we're in what we would call G0, after we've made all the cells of a particular type we're going to make, this is where we stay, right? We just stay here and do our job, and we don't divide ever again, all right? In prophase, the chromosomes condense and become visible for the first time, all right? They'll stain darkly in the nucleus, all right? They become more compact, the nuclear envelope breaks down, and the spindle microtubules get formed by the centrioles and the centrosomes, okay? And they attach to the centromeres of the duplicated chromosome. So that would be the middle of the X-shaped molecule, okay? So each side of that X is identical information in mitosis, right? The right side of the X has the same information as the left side of the X, right? Unless there's been a mutation, okay? The microtubules attach, and then they position those chromosomes in approximately the middle of the cell, all right? You can see the spindle apparatus in the microtubules latching on. And you can see the chromosomes lining up in the middle, okay? And it uses ATP to do this, all right? In anaphase, we pull them apart. So we're yanking in opposite directions. The centrioles in the centrosome are like the reel in a rod and reel. The microtubules are like the line in a fishing line. And the chromosomes are like the fish. So it's like two fishermen catch the same fish on the opposite side of the river, and they pull the fish in half. Okay. So off they go, right? So now each side only has 1/2 of that X. They're flying away. Okay. In telophase, they arrive at opposite ends of the cell. They begin to decompress, so they become less visible. And then the nuclear membrane begins to reform. And then generally that's followed by cytokinesis, right? So we form 2 new nuclei, and then we pinch the rest of the cell in half, and then you have 2 new cells, right? So you've got the same chromosome number, you got the same genetic information, and the cells are going to look and act the same as the original, okay? That's asexual reproduction. The cytoplasm divides between late anaphase and the end of telophase. 2 new cells form, each with their own nucleus. Mechanisms of cytoplasmic division differ between animal and plant cells. In animal cells, we make what's called a cleavage furrow, formed by a contractile ring that is surrounded by stuff that's similar to actin, which is what makes one of the proteins that helps make muscle contract, okay? The cleavage furrow deepens until the cytoplasm is pinched in 2. Each new cell has its own nucleus, its own cytoplasm, and its own plasma membrane, and its own nuclear membrane, right? So everybody is getting the same amount of material. They're going to look and act like the original, right? Carbon copies of each other, all right? Okay. In plant cells, it's a little bit different. Microtubules guide the vesicles from Golgi bodies and the cell surface of the division plane. The vesicles and their wall-building components fuse into a disc-shaped cell plate, which grows and forms a cross wall between the 2 new nuclei. The cell plate develops into 2 new cell walls and separates the descendants, okay? So that's different from forming a cleavage furrow. So we just build a new wall, okay? We build a new wall, and then the plasma membrane just sort of closes in on either side of the wall, and you got 2 new cells. So we don't really pinch them in 2. We just build a partition, okay? And that's going to be in any eukaryotic cell that has a cell wall, okay? A plant cell, a fungal cell, okay? All of those have cell walls. They're all going to do a plate that divides the material, okay? So those vesicles contain the cell wall material, the material deposits, the plate expands, and you got 2 new cells. Right? But how do we control the cell cycle? There's a lot of genes involved in that, right? When a cell divides, this is determined by gene expression. Proteins govern this. The products of checkpoint genes monitor whether a cell's DNA has been copied completely, whether it's damaged, and whether enough nutrients are available. If the problem remains uncorrected, other checkpoint proteins may cause the cell to self-destruct, okay? So you can see an example here of some checkpoint gene products in action. This is fluorescence microscopy. The way that we do this kind of picture is that we actually coat the material with antibodies that have a dye attached to the antibody that when a laser beam hits it, it glows a particular color, okay? So everywhere you see a spot, that's your molecule, okay? It's been tagged and it lights up, which is kind of cool, okay? So this is a look at the nucleus of the same cell that was exposed to radiation. So the DNA is damaged. BRCA1 and P53 appear in the same places in the 2 photographs because they have clustered around the same sites of DNA damage. The integrated action of these and other proteins prevents DNA replication from occurring until the DNA is fixed. And these are called repair genes, right? You bust me up because you hurt me. I got to fix what you bust up before we reproduce. If I don't do that, we're going to have a problem because we're not going to be missing material. We might have mistakes in the material. That's no good. This is one of the reasons, for instance, that when you're out in the sunshine and you got UV hitting your skin, that you make melanin and your skin tans, right? It's to protect the DNA in the cells from the damage that UV causes when it makes the DNA double helix buckle, right? Because the UV rays hit it, the DNA undergoes a chemical reaction that produces a buckle of the double helix. If we don't repair that properly, we're going to make a mutation and that could lead to skin cancer. All right. This is just showing you the repair genes in action. Okay. Tumor suppressors do what their name implies, right? Prevent tumors from forming. Checkpoint genes that inhibit mitosis are tumor suppressors because tumors form when these genes are knocked out. Okay. So you break these genes, a tumor will happen. Okay. Viruses like human papilloma cause a cell to make proteins that interfere with its own tumor suppressors that can lead to cancer. This is why, for instance, in young women, we now have an HPV vaccine, right? We try to prevent you getting infected with human papilloma because that can lead to cervical cancer. And the reason for that is the HPV can integrate in the DNA of your tissue and produce a mutation that can lead to a cancer cell being made, right? What is cancer? Cancer is uncontrolled cell growth, all right, for reasons generally of mutation. A cell cycle gene has gotten messed up. As a result, we divide in a fashion that does not serve the host, okay? In a neoplasm, we see accumulation of abnormally dividing cells. A tumor is a neoplasm that forms a lump, and an oncogene is a gene that helps turn a normal cell into a tumor cell. Genes for proteins that promote mitosis are called proto-oncogenes because mutations turn them into oncogenes. Examples include genes that code for receptors for growth factors. Okay. You can see an example of tumor progression here. Benign tumors grow slowly and stay in their home tissue. Malignant tumors grow quickly and their cells don't stay anchored to home tissue. They become attached to the wall of a lymph vessel or a blood vessel, release digestive enzymes that create an opening, enter the vessel, and travel. That's metastasis. The cells move through the vessel, exit the same way they got in. Migrating cells can start another tumor in another location. The characteristics of cancer cells, they grow and divide abnormally, meaning continuously, right? Abnormal plasma membrane, cytoskeletal, chromosome number, and metabolism are hallmarks. The cells don't stay anchored properly in tissues because plasma membrane adhesion proteins are defective or missing. So basically these guys, they get built and they go on a walk, right? And wherever they land, they do their thing. And how do we deal with this? Okay. And the answer is not that well. Okay. The options that are available for cancer treatment include immunotherapy, radiation, and chemo. All right. Except in certain cases where we've come up with drugs for particular types of cancer that are very specific for that type of cancer and don't produce damage in other tissues. All right. Examples include things like leukemia, certain types of leukemia, and retinoblastoma. All right. But most other cancers, we don't have a magic bullet like that yet. Okay. So here's a look at some cancer treatment.
If you have cancer, your doctor may recommend chemotherapy as part of your treatment. The cells in the body grow and divide as part of the normal cell cycle. The cell's nucleus controls this process. Inside each nucleus, genetic material called DNA contains the instructions for directing this process. Sometimes the cell's DNA becomes damaged. Normally, the DNA responds by either repairing itself or instructing the cell to die. In cancer, however, the parts of the cell's DNA that direct cell division become damaged. When these sections are damaged, the DNA is unable to repair itself or cause the cell to die. Instead, the unrepaired DNA causes the cell to grow and divide uncontrollably into more damaged cells called cancer cells. A tumor forms as the cancer cells multiply and displace the normal cells. As the tumor enlarges, it develops its own blood supply. Since cancer cells do not stick together as well as normal cells, they may break away and enter a nearby blood vessel. Cancer cells in blood vessels may travel to other areas of your body and form additional tumors. This is called metastasis. Additional tumors may form in areas such as the lungs, liver, and bones. Another way cancer may spread to other areas of your body is through your lymphatic system. Cancer cells may enter lymph vessels near the tumor, then travel to small glands called lymph nodes. If the cells pass through the nodes, they may continue to travel through your lymphatic system and form additional tumors. Chemotherapy drugs work by targeting fast-growing and reproducing cells, a characteristic common to cancer cells. The tumor shrinks as the cells stop dividing and die. Most chemotherapy drugs work systemically as they travel throughout your body in your bloodstream. As they circulate, the drugs damage metastatic cancer cells in other organs. Unfortunately, chemotherapy drugs cannot tell the difference between fast-growing normal cells and cancer cells. As a result, these drugs also damage or irritate some of your fast-growing normal cells, such as those in your bone marrow, digestive system, and hair follicles. Death, irritation, or damage to these normal cells produces side effects such as a weakened immune system, nausea, and hair loss. The goal of chemotherapy is to reduce or eliminate cancer cells in the original tumor and any sites of metastasis. In addition to being a primary cancer treatment, doctors often use chemotherapy as a secondary treatment before, during, or after other primary cancer treatments, such as radiation therapy or surgical excision of a tumor. Depending on the location and type of cancer, you may receive chemotherapy drugs intended to circulate throughout your body, including pills, capsules, or liquids taken orally, and intravenous or intramuscular injections. Alternatively, you may receive drugs delivered only to the area of the tumor. One local method delivers drugs to your bladder or chest through narrow tubes called catheters. Another local method injects drugs into the cerebrospinal fluid surrounding your brain and spinal cord. A third local method places slowly dissolving wafers into an area where a tumor was removed. In most cases, you will receive a number of different chemotherapy drugs to increase their effectiveness. You may receive many chemotherapy treatments spread out over a period of weeks or months. This allows your body to recover between treatments and to kill as many cancer cells as possible. Common side effects of chemotherapy include hair loss, nausea, decreased appetite, fatigue, anemia, bruising, and diarrhea. It is important to rest, eat nutritious foods, and take medications prescribed by your doctor to reduce or minimize these side effects.
And this is radiation.
Your doctor may recommend intensity modulated radiation therapy, a procedure that treats certain forms of cancer to kill cancer cells or control their growth. The cells in the body grow and divide as part of the normal cell cycle. The cell's nucleus controls this process. Inside each nucleus, genetic material called DNA contains the instructions for directing this process. Sometimes the cell's DNA becomes damaged. Normally, the DNA responds by either repairing itself or instructing the cell to die. In cancer, however, the parts of the cell's DNA that direct cell division become damaged. When these sections are damaged, the DNA is unable to repair itself. Instead, the unrepaired DNA causes the cell to grow and divide uncontrollably into more damaged cells called cancer cells. A tumor forms as the cancer cells multiply and displace the normal cells. Radiation kills cancer cells by inflicting overwhelming damage to their DNA. The tumor shrinks as the cells stop dividing and die. If the tumor is in your head, your radiation therapist may make a mesh frame or mask that will keep you positioned correctly during the IMRT. Or you will get small permanent tattoos to help align the IMRT. Next, your therapist will do a CT scan of your head to get a detailed picture of your tumor and its borders. Your doctor will use the scans to specify the three-dimensional shape of your tumor and vital tissues nearby. The tumor's shape, size, type, and location help your doctor determine how the IMRT beam should be adjusted to hit your tumor and avoid healthy tissue. During your procedure, a medical linear accelerator will deliver an X-ray beam to your tumor. A computer inside the accelerator will adjust the beam with a device called a collimator. The collimator will adjust or modulate the intensity, direction, and shape of the radiation beam. This feature allows higher doses of radiation to be delivered to your tumor while sparing your healthy tissue around it. You may receive many radiation treatments over days or weeks. This allows your body to recover between treatments and to kill as many cancer cells as possible. Side effects are usually mild and occur only in the treatment area. Common side effects include localized skin irritation, localized hair loss, or inflammation and swelling near the tumor location.
Now, that said, right, one of the outcomes of these two types of treatment is the production of mutation in healthy tissue. Why? Radiation makes mutations, and so do chemotherapeutic drugs. So you may end up treating your initial cancer and end up with another type of cancer as a result of the treatment. So it's not the best option. There really aren't a lot of good options. Okay, it's the best of the bad situation. There are diseases that are like that. Okay, the hope is that you kill the cancer cell before you kill the patient. It doesn't always work. The easiest cancers to kill are the ones that grow the most rapidly. Why? Because they're constantly in DNA production and protein synthesis mode. And if you hit something while it's in the process of construction, you damage it more intensely, right? So paradoxically, the most rapidly growing cancers are the easiest to wipe out. The slowest growing ones are the most difficult to wipe out because they're not always in building mode, okay? You're also going to hit tissues that aren't cancer tissues. Other tissues that divide rapidly include epithelial tissue and connective tissue and so on. And so you lose the lining of your gut, your crit falls, your hair falls out, your skin thins, and you become relatively sick before you get better. There's no way around that, okay? Immunotherapy is different. In immunotherapy, what we do is we try to ramp up the immune system so that it sees the cancer and kills it independently of trying to damage the cells themselves. And sometimes it works and sometimes it doesn't, okay? What we're trying to get to now is to figure out the specific mutation that causes your particular cancer and try and find a treatment that's most successful in those types of events, And so we're getting more towards a personalized kind of cancer treatment, but we're not there yet. Okay. And then we've talked earlier about, where do mutations come from? And the fact is they happen every day, right? DNA replication is not perfect. So you get mutations every day. You get cancer 2 or 3 times a day, and your immune system wipes it out, right? You get it from viruses. You get it from mutagenic agents. You get it from exposure to radiation, okay? All of those things can make mutations. All of those things can produce cancer, right? It's just a matter of interacting with the environment, period, okay? One of the things that we notice about cancer cells is that they're immortal. As long as you feed them, they keep going. And one of the reasons for that is the telomerase phenomenon. We talked about in DNA replication, the fact that when you get to the end of the DNA molecule on the lagging strand, you can't complete the tip unless you bring in a special enzyme called telomerase that has its own primer, and you finish that little stretch of DNA at the tip, and then you recover that bit of information. If you don't do that, you lose that information. And if that happens over many, many cell divisions, eventually you get to critical information, the cell dies. But in cancer cells, frequently the telomerase is overexpressed. And that's one of the genes that makes you immortal. Okay. So under normal circumstances, eukaryotic chromosomes shortened by about 100 nucleotides per replication cycle. Too short of a telomere triggers a checkpoint that halts cell cycle and cause death, right? We talked about, again, why we die, right? It's the integrity of the information to a large extent. Paired genes on homologous chromosomes vary a little bit with DNA sequence, and those are like flavors of a gene. Alleles are forms of a gene that encode slightly different versions of a gene product. Offspring of sexual reproducers inherit new combinations of alleles, which is the basis of the combination of new traits. And that's because of, again, good old recombination at prophase of meiosis 1, right? Homologous chromosomes come together, they exchange genetic information in areas where they're synapsed, and then about 50% of the DNA is what's called recombinant, rearranged, right? And so you've got, for every sperm cell, every egg cell that you make, right, about half of that genetic material is rearranged. And the result is biodiversity in the population, right? And that's the advantage, right? In asexual reproduction, everybody's a clone of everybody else. In sexual reproduction, you have biodiversity. And so when you have a challenge to an asexually reproducing population, everybody's going to suffer because everybody's the same pretty much, okay? Whereas if you have a challenge to a sexually reproducing population, it's not likely to wipe out everybody. There's going to be some that survive because they have characteristics that help them to live long enough to breed, because everybody's a little bit different, okay? So that's a good thing, all right? So you can see examples, right? On the right there, you've got asexual reproduction. That is, I believe, a mother of millions plant. You guys were asking me about that earlier. See the little plant sproutlets on the edge of the leaves? Those are clones of the original plant. When they fall to the ground, they just grow, okay? They're all ready to go. And on the left, of course, you've got sexual reproduction with a dog, right? So the disadvantage, of course, is in asexual reproduction, everybody's the same, okay? It's rapid, but there's no diversity except for the diversity produced by mutation, which is much lower than diversity produced by bringing together mom and dad's genes, right? And recombining some of that stuff. Meiosis, we talked about earlier, right? In meiosis, what happens is that you generate from an original diploid cell 4 haploid cells that have half the amount of genetic material as the original. And then in fertilization, you restore the diploid number and make a new organism, okay? In a human female, for every oogonia that enters meiosis, you get 1 egg. And in men, for every spermatogonia that enters meiosis, you get 4 sperm. Now, the reason for that is that in meiosis 1 and meiosis 2, in mammalian females, you lose one of the 2 products of cell division as what's called a polar body, so that the cytoplasm doesn't divide equally, and one of those products just degenerates and doesn't do anything, okay? So at meiosis 1, in a female, when the oogonia divides, you get 1 cell that's going to go on becoming a primary oocyte and one polar body. And then in meiosis two, you do the same thing again. That one good cell divides, you make another polar body, and you end up with one secondary oocyte that becomes the egg. Okay. But in a male, the spermatogonia divides once for meiosis one, and then each of those divide once for meiosis two, and you end up with 4 sperm cells, right? So guys crank out way more gametes than women do, okay? And they do it from puberty until death, right? Whereas a woman is born with all the eggs she's ever going to have, and as she ages, her eggs also age, okay? And they don't finish meiosis until what? They're ovulated and fertilized, right? The egg has to pop out of the ovary, and a sperm cell has to hit it. And once the sperm cell hits the surface of the egg, it real quick finishes meiosis two, makes that next polar body, and then the sperm cell and the egg cell nucleus fuse, and that's your zygote. You're ready to go, okay? But until then, what's it doing? It's in suspended animation, waiting for those two events to happen. And the longer it waits, the more likely it is to make a goof, and that goof can lead to what we call aneuploidy, meaning an improper chromosome number that can lead to birth defects or even spontaneous abortion, okay? And so that's why a lot of women are told if you want to have kids, you want to do it before 35, okay? Because after 35, the odds of that happening go way up, all right? Why? Errors in the spindle apparatus, problems with chromosomes linking together in a way that you can't pull them apart, okay? The longer they sit having exchanged genetic material, the harder it is to finish the job, okay? You can see here how it works, right? In meiosis two, we have what we call the equational division. In meiosis one, we have what we call the reductional division, okay? So in meiosis one, we go from the diploid to the haploid state. And in meiosis two, we remain in the haploid state, but we end up at the end of the day with 4 products that have half the amount of genetic material as the original, right? Meiosis recombines alleles on homologous chromosomes through a process of crossing over, which happens in prophase of meiosis one, and then random assortment scrambles up the traits even more, okay? The way a chromosome lines up on the metaphase plate as a homologous pair is a random event, all right? In crossing over, you can see that we can switch out versions of a gene from one chromosome to the other chromosome. They find each other based on sequence similarity, right? So dad's chromosome 1 finds mom's chromosome 1 because they're pretty similar, but not identical, right? And then they swap little bits of genetic material in what's called a stochastic process, a random process, right? Where do we cross over? It depends, okay? It's random, but there are some spots in the DNA that seem to be crossover hotspots, right? But you can see, right? We flip and swap, and then we get, on average, about 50% of the genetic material when we're all said and done being recombinant, meaning unlike what's found in either parent, okay? And that increases biodiversity, okay? So you can see here, right, after the crossing over has occurred, we've got an arrangement of genes that isn't present in either mom or dad, okay? Sexual reproduction involves the fusion of the sperm and the egg. All the gametes are haploid, and so when they fuse together, they make the diploid zygote that goes on to produce the organism, right? So that's what it is about sexual reproduction, right? So points to ponder, right? Why is it difficult to design a drug that will stop cell division only in tumor cells? Right? How does it relate directly to common side effects of cancer treatment? We kind of talked a little bit about that already, okay? What are some ways that cell division and fertilization improve genetic diversity in each generation, right? And how sexual reproduction gives you more biodiversity than asexual reproduction, or in bacteria, binary fission, right? Where biodiversity in asexual reproduction is just a result of the mutation, right? But in sexual reproduction,